Hi friends, welcome to today's episode of KPS Bio Tutorials. Today I would like to talk about the translation in prokaryotes part 3. Translation in prokaryotes part 1 ഉം part 2 ഉം കേട്ടതിനു ശേഷം മാത്രം നിങ്ങൾ ഈ വീഡിയോ കാണുക. Then only you can understand well about the translation in prokaryotes. So I will be giving the link of part 1 and part 2 in the description box. So you first listen to part 1 and part 2, then you will listen to the part 3 of the translation in prokaryotes, so that you can understand well the subject. So today I would like to talk about the initiation of translation. You know, in transcription, translation and replication, all these processes are taking place in 3 steps: initiation, elongation and termination. Translation എന്ന് പറഞ്ഞാൽ നമുക്കറിയാം, it is the incorporation of amino acids according to the codons in the mRNA. mRNAയിലുള്ള കോഡോൺസിന് അനുസൃതമായി അമിനോ ആസിഡുകളെ പെപ്റ്റൈഡ് ബോണ്ട് വഴി ലിങ്ക് ചെയ്ത് ഒരു പോളിപെപ്റ്റൈഡ് ചെയിൻ ഫോം ചെയ്യുന്നതിനാണ് നമ്മൾ ട്രാൻസ്ലേഷൻ അല്ലെങ്കിൽ പ്രോട്ടീൻ സിന്തസിസ് എന്ന് ഉദ്ദേശിക്കുന്നത്. So initiation of translation means it is the whole step of translation up to the incorporation of first amino acid in the polypeptide chain. സിന്തസിസ് ചെയ്യാൻ പോകുന്ന പോളിപെപ്റ്റൈഡ് ചെയിൻ്റെ ആദ്യത്തെ അമിനോ ആസിഡിനെ ഇൻകോർപ്പറേറ്റ് ചെയ്യുന്നത് വരെയുള്ള പ്രോസസ്സുകളെയാണ് നമ്മൾ ഇനീഷ്യേഷൻ എന്നതുകൊണ്ട് ഉദ്ദേശിക്കുന്നത്. കഴിഞ്ഞ എപ്പിസോഡുകളിൽ നമ്മൾ ചർച്ച ചെയ്തിരുന്നു, translation has many requirements like ribosome, tRNA, amino acid tRNA synthase enzyme, translational factors, etc. You know, initially the ribosomes, you know, the prokaryotic ribosome is the 70S ribosome, which contains 30S subunit and 50S subunit. These two subunits will be in the separated form before the initiation of the translation. The translational factors IF1 and IF3 will be bound with the 30S subunit, so that the 30S subunit cannot bind with the 50S subunit. Thereby it will be in separated form. So if there is no translation, the ribosomal subunits are in the separated form. The initiation of the translation actually having 2 steps. One is the binding of mRNA with 30S subunit. The other one is the binding of the initiator tRNA. That means the tRNA which will bring the initiator amino acid. Mostly the initiator amino acid is methionine. So the initiator tRNA with the initiator amino acid methionine will bind at the P site of the 30S subunit. This is the second step in the initiation of translation. Then after that, the large subunit will bind on the 30S subunit, or the complete ribosome will be formed. So all the processes up to the formation of the 70S or the complete ribosomal complex is said to be the initiation. You know, before the translation begins, the 30S and 50S subunit will be in separated form, because the translational factors IF1 and IF3, that is initiation factor 1 and 3, will be bound with the 30S subunit. Thereby it cannot bind with the 50S subunit to form the complete 70S ribosome. So the function of the initiation factors 1 and 3 is to prevent formation of complete ribosome before the initiation of the translation, or before the binding of mRNA and initiator tRNA. So only after the binding of initiator tRNA and mRNA with the 30S subunit, the 50th subunit can bind with the 30S subunit to form the complete 70S ribosome. The first step in the initiation of translation is binding of mRNA with 30S subunit. The binding of mRNA with 30S subunit occur by the base pairing of the mRNA, a particular sequence in mRNA, namely Shine-Dalgarno sequence, which is located a few base pairs upstream of the start point. The start point in mRNA is the first codon, that is AUG. Upstream, a few base pairs, about 10 base pairs upstream of this start codon in the mRNA, there is a special sequence, namely Shine-Dalgarno sequence. This Shine-Dalgarno sequence will base pair with its complementary bases in the 16S rRNA in the 30S subunit. You know, the 30S subunit contains the 16S rRNA and many R proteins. The rRNA present in the 30S subunit is 16S rRNA. This 16S rRNA contains complementary base pairs to the Shine-Dalgarno sequences in the mRNA. So this base pairing between Shine-Dalgarno sequence and 16S rRNA enable the mRNA to bind with the 30S subunit. Here you can see the Shine-Dalgarno sequence, which is located 10 base pairs, about 10 base pairs upstream of the AUG start codon, and which will make complementary base pairing with the 16S rRNA. Here there is Shine-Dalgarno sequences of different strains of E. coli and bacteriophages. They are located upstream of the start codon AUG, and usually the sequence of Shine-Dalgarno sequence, usually the base sequence of Shine-Dalgarno sequence is AGG. Its number may vary from 3 to 9. The number of Shine-Dalgarno sequence may vary 3 to 9 base pairs, and this Shine-Dalgarno sequence is purine rich. The next step of initiation of translation is binding of tRNA F at P site. You know, the tRNA binding at P site, or it is the initiator tRNA, is a special tRNA. So it is called tRNA F. Here F indicates the formyl tRNA, because the amino acid linked to tRNA F will be formulated. So the initiator tRNA is said to be tRNA F. The initiator amino acid is methionine. So the methionine bound to the tRNA F will be formulated, and this formulated methionine can only bind with the P site. So the starting or initiator tRNA is said to be tRNA F, and initiator amino acid is said to be F formulated methionine. So the next step is the amino acylation of the initiator tRNA with methionine amino acid. A methionine amino acid will bind with tRNA F in the presence of, with the help of the enzyme amino acyl tRNA synthetase enzyme. This is the enzyme which will make phosphodiester bond between the methionine and tRNA F. It is the enzyme which catalyzes the formation of an ester bond between methionine and tRNA. Thereby, a methionine tRNA F will be formed. Then the methionine acylated with the tRNA F is formulated. This formulation occurs with the help of N10-formyl tetrahydrofolate. That means a formyl group from the N10-formyl tetrahydrofolate is given to methionine tRNA-F, or methionine amino acid bound to the tRNA-F. Thereby N10-formyl tetrahydrofolate become tetrahydrofolate, and thereby methionine tRNA-F, or the methionine on the tRNA-F, become formyl methionine. So at the end of this reaction, a compound, namely N-formyl methionine tRNA-F, will be formed. This N-formyl methionine tRNA-F can only be inserted to the P site in the 30S subunit of the ribosome. And this N-formyl methionine tRNA-F compound will be transported to the 30S subunit by the initiation factor 2. The initiation factor 2 will bind with GTP, and thereby a conformational change will occur on the IF2. This conformational change enables the IF2 to associate with the formyl methionine tRNA-F, and the IF2 in the GTP form will bind with the N-formyl methionine tRNA-F, and which will bring the N-formyl methionine tRNAF to the 30S subunit. So the function of IF2 is to transport the N-formyl methionine tRNAF to the site of protein synthesis or to the P site of the 30S subunit of 70S ribosome. After entering the N-formyl methionine tRNAF or the tRNA initiator tRNA with the initiator amino acid, it will read the codon, the initiator, the anticodon in the initiator tRNAF or tRNAF molecule will read codon in the mRNA. The starting codon is the AUG, mostly it is AUG, and in some cases it is GUG. If the codon is AUG, at the initiation site it code for formyl methionine, and if it is GUG, at initiation site it code for formyl methionine, and if it is GUG, at the initiation site it also code for formyl methionine, and at intervening sites the AUG code for normal methionine and GUG code for valine amino acids. So the anticodon in the tRNA will make complementary base pairing with the codon in the mRNA. If this anticodon-codon interaction is stable, that means if it is the exactly base pairing, the codon is reading correctly with the anticodon, then the GTP bound to the IF2 undergo hydrolysis, and thereby the other initiation factors IF1 and IF3 will be released from the 30S subunit. If the codon-anticodon interaction is stable and correct, the initiation factors IF1 and IF3 will be released from the 30S subunit by the hydrolysis of GTP bound with the IF2. If the codon-anticodon interaction is suitable and correct, the GTP bound to the IF2 undergo hydrolysis, and which cause the release of the initiation factors IF1 and IF3 from the 30S subunit. As a result, now the 30S subunit is free to bind with the 70S subunit. Now the 30S subunit is free to bind with the 50S subunit. Thereby the 50S subunit comes and bind with the 30S subunit, so that the complete 70S ribosome will be formed. Now this whole complex is said to be 70S initiation complex, which now contain the complete 70S ribosome, mRNA, and the initiator tRNA with initiator amino acid methionine. This entire complex is said to be 70S initiation complex. The whole set of reaction of translation up to the synthesis of this 70S initiation complex is said to be the initiation of translation in prokaryotes. So I think all of you understood well about our initiation of the translation. If you have any doubt, you may comment in the comment box. And if you like this video, or if you understood well about the subject, you will like the video and you may share with your friends. Also, if you are not subscribed the channel, you subscribe the channel. Also enable the bell icon so that you will get the notification when I upload the videos. So thank you very much for watching the class. Once again, thanks to all.